Endochondral ossification produces bone from pre-existing hyaline cartilage.
The bone collar is produced when the perichondrium, the outer edge of the developing bone, begins producing osteoblasts. The collar provides structural reinforcement. In this stage, primary ossification occurs as chondrocytes in the center of the model enlarge and the cartilage matrix begins to deteriorate.
At this point, blood vessels penetrate the bone collar. The primary marrow cavity is formed as the center is hollowed out. Here, osteoid tissue calcifies to form temporary trabeculae. Cartilage dies in the diaphysis and is transformed to bone by osteoclasts.
Now, secondary ossification sites appear in the epiphyses. These sites hollow out and undergo the same transformation as the primary ossification center. In humans, this stage usually corresponds with birth. The joints are still cartilaginous. During infancy and early childhood, spongy bone develops in the epiphyses, at which point cartilage only exists in joints as articular cartilage and in the epiphyseal plate. The epiphyseal plate is the location of bone growth and is often referred to as the growth plate.
By the late teens to early 20s, all remaining cartilage in the epiphyseal plate is generally consumed and the gap between the epiphyses and diaphyses closes. The primary and secondary marrow cavities then unite into a single cavity, and the bone can no longer grow in length.
Intramembranous ossification produces flat bones, often called dermal bones, within a fibrous sheath. Some dermal bones include the clavicle and parts of the skull. First, an ossification center appears in the fibrous connective tissue membrane. Then, bone matrix is secreted within the fibrous membrane. Later, woven bone and periosteum form. And finally, the bone collar of compact bone forms and red marrow appears.